Hello guys, my name is Kirtan and welcome to my channel Physiology at Once. Today I will be talking about the topic Physiology of Immunity. This is the first part of this topic. In this video I will be talking about innate immunity. So let's start. What is immunity? It is the process of defense system of the body against harmful elements which threaten our normal health. So anything which harms our body, it is defended by our immunity. Largely immune responses can be divided into two types, innate immunity and acquired immunity. So today we are talking about innate immunity. Under the innate immunity, it can be divided into mechanical defense, chemical defense, defense by natural killer cells, defense by phagocytic cells, defense by inflammation and fever. This innate immunity can also be called as non-specific defense system because this immunity is not specific to a single antigen. So let's start the mechanical defense. In the mechanical defense, they are again divided into skin and epidermis. Skin and epidermis are majorly involved in the mechanical defense. So during burns and septicemia, there is major cause of infection because there is loss of skin and epidermis. Mucous membrane. This mucous membrane helps in protecting the body where there is no skin or epidermis. Mucus. Mucus is secreted by the mucous epithelial cells and excretes many waste substances through it. Hairs. Hairs are present in almost all the opening and it also traps the microbes coming through the air or water. Cilia. It also has the similar function as hairs and is present in the openings, or it is also present in the upper respiratory tract. Tears. It is produced through the lacrimal apparatus and clears the microbes and has acidic properties which reduces the microbial activity. Saliva. Saliva also has the acidic properties which destructs the microbes. Urine. Through urine many toxic materials are released and it flushes out the urinary tract. Defecation. Through defecation also many toxic are released. Chemical defense. Acidic pH of skin. Skin is actually little acidic which helps to clear the microbes out of it. Bactericidal substance of sebum. Sebum actually contains unsaturated fatty acids which help in destructing the microbes. Chemicals in body secretions. In the body secretions such as saliva, gastric secretion, there are many chemicals which react with the microbes and kill it. Acidic pH of stomach and vagina. The pH inside the stomach is acidic due to the HCl present in it, which kills the microbes coming through the food. Complement proteins. These complement proteins are produced through the complement system of the immunity. Interferons. Interferons are produced from the activated lymphocytes and also helps in the killing of microbes. So the next one is defense by natural killer cells. Natural killer cells are the third category of lymphocytes. They differ from other lymphocytes by they do not require prior sensitization to kill microbes, whereas the other lymphocytes require some prior sensitization, like it needs to be activated to kill microbes. The major histocompatibility complex or MHC antigen is not required, whereas the other lymphocytes require it. They are the first line of defense against viral infections. So these natural killer cells are increased greatly during the viral infections. They are also active against tumor cells, especially malignant cells. So they are greatly increased during viral infections and tumor cells. The mechanism of killing of the natural killer cells. First, osmotic lysis occurs by incorporating perforins into the surface of the microbes. So when the perforins attach to the surface of the microbes, it makes little holes which lyses the cell wall of the microbes. Then there is release of interferons that activate the phagocytosis and immunity. These interferons act as the communicating signal which activates the phagocytes and activate the phagocytosis process. They possess the FC receptors. The natural killer cells possesses the FC receptors that allow them to kill antibody-coated viruses. They kill by the process of ADCC. ADCC is also known as antibody dependent cell-mediated cytotoxicity. This is a cytotoxic process through which especially virus and tumor cells are lysed. So now let's talk about the defenses by the phagocytosis cells. They contain both granulocyte and agranulocyte in the phagocytosis process. As I had told before in the WBC video, the phagocytosis occurs through chemotaxis, margination and adherence, ingestion and killing. You can check the video of WBC and I'll put that in the I button link. The main phagocytes are neutrophils, monocytes and the macrophages. The macrophages can also be again divided into fixed macrophages and wandering macrophages. Under the fixed macrophages, it forms the mononuclear phagocyte system, and under that it comes Kupffer cells of liver, alveolar macrophages in lungs, microglia of brain, osteoclasts in the bone, mesangial cells in the kidney, histiocytes in the connective tissue, Langerhans cells in the skin. So overall, this mononuclear phagocytosis system is present all over the body. So this one is very important and you need to remember this because this can be asked in the exam. So now let's talk about the defense by inflammation and fever. In inflammation, it is the defensive response of the body against the tissue injury. It is characterized by 4 features: rubor, which is known redness, dolor or pain, calor or increased temperature, or tumor, which is also known as swelling. You can remember this by red dog chases a train. Red dog chases a train. So these 4 characteristics are important under the topic of inflammation. The stages of inflammation are, the first one is vasodilation and increased permeability. In this there is production of histamine, bradykinin, kininogens, prostaglandins and complement proteins. Histamine is produced by the mast cells. Kininogens are the tissue kininogens and plasma kininogens. Prostaglandins are produced by the damaged cells. And complement proteins are produced by the complement system. This bradykinin is present in the blood. And then the contraction of vascular endothelial cells also facilitates the vascular permeability. So there is contraction of vascular endothelial cells also. Then the arterial dilation increases further blood flow that causes increased local temperature and flare. The next step is phagocyte migration and phagocytosis. Within some time, phagocytes appear at the site of injury in the order neutrophil, monocyte and macrophage. It means that neutrophil comes first, then the monocyte and then the macrophage. So therefore, during the infection time, the neutrophils are increased. During the inflammatory response, phagocytes die and form viscous fluid which is called as pus. The next step is tissue repair. It is facilitated by migration of fibroblast, macrophages and epithelial cells to the site of injury that repairs and restores the epithelium by secreting growth factors. The next one is fever. The features of it are there is increased body temperature which prevents the growth of microorganisms. It also facilitates the action of interferons and enzymes which inhibit the growth of any other microorganisms. As I told before, interferons acts as communicating signals. So it activates the lymphocytes. So that's it for today guys. If you like this video, please like, share and comment and also subscribe for more videos like this.